Hi everyone, this is part 2 of our chapter 3 lecture for your Marib text. Chapter 3 covers cells. Okay, so in part 1 we talked about passive membrane transport. So the second half of this chapter we're going to be covering active membrane transport. So if you remember, passive transport was transport that relies on the movement of molecules due to their concentration gradients or just diffusion, right? So molecules tend to move from areas of high concentration to low concentration, and that does not require any energy. Active membrane transport is the other type of membrane transport, and it's both the active membrane transports. There's two different types. Active transport and then vesicular transport. Both of these are types of active membrane transport, and they're classified that way because they both require ATP or energy to move solutes across the plasma membrane. And they need a lot of energy for a few reasons. So one, the solute is too large for channels, the solute is not lipid soluble, or the solute is not able to move down its concentration gradient, which means the solute is moving against its concentration gradient. So instead of going from an area of high concentration to low concentration, active transport uses energy to pump solute molecules against their concentration gradient, so a way they don't want to naturally go. All right. So active membrane transport is always going to require some sort of carrier protein because the molecules aren't just going to diffuse through the plasma membrane themselves. Never going to happen. So these carrier proteins, like the ones we talked about in facilitated diffusion, are going to bind specifically, which means that each carrier molecule only binds to specific molecules. And it also binds reversibly. So we didn't really talk about this too much with facilitated diffusion, but after a carrier protein moves a molecule, it's able to move more and more and more. It typically does only one or a couple at a time. Okay, so carrier molecules, again, are binding specifically to specific molecules, but some are able to bind to more than one type of molecule. But again, it's going to be specific. Not everything can bind to it. So there's two types of these carrier proteins that we're going to talk about. We have antiporters and symporters. Okay, so antiporters are going to transport two different things. So it's going to transport one substance into the cell while transporting a different substance out of the cell. Okay, so it's going in opposite directions. So antiporter, I'm just going to write here, they're going in opposite directions. Okay, symporter also is going to move two different substances, but they're both going to be going in the same direction. Okay, so both molecules are either going to be going into the cell or both molecules are out of the cell. So antiporters opposite, symporters are going to be going moving molecules the same way. Okay, and for active transport, again, we're going to be moving solutes against their concentration gradient. And because we're moving them against their concentration gradient, we're going to need energy or ATP to move them. Okay, again, we can't just use diffusion, let the molecules move on their own. We have to take energy and pump these molecules out. Okay, and there's two different types of active transport. We have primary active transport. Hold on. Stella, come here. Stella. No. Sorry, my dog saw someone outside. We have primary active transport, which uses energy directly from ATP hydrolysis to move molecules. And secondary active transport, which also uses energy, but the energy is obtained indirectly. Okay, so I'll give you some examples in a minute, but for secondary active transport, typically it's going to be moving molecules from ionic gradients that have already been created by primary transport. So primary transport uses energy from ATP directly. Secondary active transport uses the results of the primary transport to move additional molecules. Okay, and I'll give you an example, and I think it'll make a little bit more sense. Okay, so first let's talk about primary active transport. So like I said, primary, you're using ATP energy directly, and basically what happens is the ATP allows the carrier protein to change in shape. The shape change causes solutes. A lot of times you're going to see ions bound to the protein to be pumped across the membrane. Okay, so there's different pumps, we call them, because they're pumping them against their concentration gradient. Calcium pumps, hydrogen or proton pumps, and the big one we're going to be talking about this week and also in future lectures are sodium-potassium pumps. Okay, and again, they're pumping these ions against their concentration gradient and using energy as a means to do that. Okay, so a little bit more about the sodium-potassium pump. This one is the most studied pump in, anatomy, physiology. It's located in all plasma membranes, but especially cells that are excitable. So muscle cells and nerves. You're going to see a lot of sodium-potassium pumps, and their activity is going to be really important. Okay, so sodium-potassium pumps are an antiporter, right? Which means it pumps one thing in one direction and the other thing in the opposite direction. So it's going to pump sodium out of the cell and potassium into the cell. Okay, remember, antiporter, opposite. So sodium goes out, potassium comes in. And this is a primary active transport. So this means it is going to directly require ATP energy to do this. Okay. Sodium-potassium pump is really important because it maintains an electrochemical gradient, which involves both concentrations of ions and the charge of ions. And you'll see as we get further into, we're talking about muscles and the nervous system, this electrochemical gradient is what's going to allow our cells to be able to send signals and communicate and, tell muscle cells to contract. And for muscle cells to actually contract, they need this electrochemical gradient and the movement of ions in order to do so. Okay, so this picture, for some reason, is a little bit blurry. But the sodium-potassium pump, again, an antiporter. So you see it's using energy here. So you have sodium. It's hard to see, but 3 sodiums are going to bind in this antiporter. ATP is hydrolyzed, okay, which is going to create an ADP in a phosphate. That's going to cause a conformational change in the shape. So you see now the shape went from a downwards V to an upwards V. So when it changes shape, the sodium ions are going to be released into the extracellular space. Once the ion pump is open that way, now potassium is going to come in and bind. And then potassium, again, we're going to change shapes again back to its resting shape. Potassium is going to be released into the intracellular fluid. And then you're going to have ATP bind again, and the same thing happen over and over again. Okay, so this is a very common example of primary active transport, and we'll see this again throughout the semester. When we get a little bit further on, it's going to be really important for you to understand exactly what the sodium-potassium pump is doing. But for now, you should just know that it's an antiporter, meaning it's pumping ions in different directions. And maintaining that electrochemical gradient. Okay, so that's primary active transport. Okay, secondary active transport doesn't directly use the energy, rather it uses the ion gradient that was created by the primary active transport system. Okay, so the sodium-potassium pump, pumping potassium out, potassium in, sodium out, It's making this concentration gradient, so making potassium very highly concentrated inside of the cell and sodium very concentrated outside. Okay, so potassium has a high concentration in, sodium has a high concentration out. Okay, and that concentration gradient is created by the primary transport system. Okay, because the primary active transport pump used electrical energy through ATP to create this concentration gradient, that now can drive transport of other solutes. Okay, so now we have a high concentration of sodium outside of the cell due to the sodium-potassium pump, right? You'll see in secondary active transport, sodium can drag other molecules with it as it flows through the cell carrier proteins into the membrane. Okay, so sodium is highly concentrated outside the cell due to energy, sodium-potassium pump. That makes sodium want to diffuse back into the cell, and when it does that, it's going to carry other things with it. Okay, and that's secondary active transport, because without primary active transport, you wouldn't have this concentration gradient. So you have to set up the concentration gradient first, and then secondary active transport uses that concentration gradient to move other stuff. Okay, so here is an example of that. So we have our sodium-potassium pump. Okay, so we have a lot of sodium outside the cell because it pumps 3 sodium out. It's going to pump 2 potassium in to the cell, creating that concentration gradient. So that's primary, directly using energy. Secondary active transport, now that we have a concentration gradient, sodium is going to want to diffuse back across the membrane. Okay, and there's membrane co-transporter proteins that it's going to go through. And as sodium wants to come in, other things are going to follow it. So in this example, we have glucose molecules that normally wouldn't enter the cell, right? But with this secondary active transport, as sodium's coming in, glucose is going to come in with it. Okay, and it drives glucose against its concentration gradient into the cell. So again, even though we're not directly using energy, we're using this electrochemical gradient that we created with sodium and potassium to drive that glucose against its concentration gradient into the cell. Okay, so active transport, we're always going to go against the concentration gradient, right? Whereas passive transport, we always went with the concentration gradient and used diffusion. Okay, so the difference again between primary and secondary: primary uses ATP, secondary uses the concentration gradients created by primary to drive stuff against its concentration gradient. Okay, so it's pretty cool. And there's a lot of different pumps that can pump things with primary active transport, and a lot of different mechanisms for secondary active transport too. Okay, so it's not just this one instance. You're going to see this with a bunch of different integral proteins that can do anti-porting or symporting using those concentration gradients to pump a number of things against their concentration gradient. Okay, so that's pretty cool. Okay, another type of active transport is vesicular transport. And it uses vesicles, that's not how you spell it, but vesicles, to move large particles, macromolecules, and fluids across the membrane in, here it is right here, in membranous sacs called vesicles. Okay, so this requires cellular energy. Again, it's an active. Right, this is also active transport, so that's usually in the form of ATP. And there's a few different types of vesicular transport, and we'll have slides on all of these. But we have endocytosis, transporting things into the cell, and exocytosis, transporting things out of the cell, and different kinds of each, so phagocytosis, pinocytosis, and receptor-mediated endocytosis. And we also have vesicular trafficking, which isn't moving things in or out. Rather, it's transporting things from one area or one organelle of the cell to another. So just moving things around in the cell itself. Some of our cells are really big. Our neurons are as long as our arms, right? We have neurons that start at our spinal cord and extend all the way to our fingertips, right? So it requires a lot of this vesicular trafficking or moving substances within the cell itself. Okay, so this is kind of what different types of the vesicles look like. So the vesicles, again, are just these membranous sacs. So they can bring things in, endocytosis, they can do exocytosis and release things back out of the cell. But in all of these, you're going to have these vesicles. And again, you're using energy to make this happen. Okay? Stella! So endocytosis, the three different types. The first type is phagocytosis, which is a type of endocytosis referred to as cell eating. Stella! Okay, so you have membrane projections called pseudopods form and flow around the particles that are being engulfed, which form the vesicles that go into the cell. Okay, so once the vesicles are in the cell, now it becomes called a phagosome. So phagocytosis usually is going to be used by macrophages or other white blood cells. So it's going to be really important in immunity and killing microorganisms or different pathogens. Okay, so it has these, again, certain receptors, those pseudopods that are going to engulf whatever it is that they're engulfing. Okay, so that's a type of endocytosis, right? Bringing things into the cell. Pinocytosis is another type of endocytosis. This is referred to as cell drinking or fluid phase endocytosis, which kind of tells you that it's bringing extracellular fluid and dissolved solutes into the cell. Okay, so it's going to be bringing a bunch of extracellular fluid from outside in. Okay, so a lot of these cells use it to sample their environment, so determining, what concentration of some substance is outside. It's the main way in which nutrient absorption occurs in the small intestine, right? So in our small intestine, as we're digesting food, our epithelial cells lining the small intestine are going to be using pinocytosis to take nutrients in. And once these vesicles are formed and the nutrients are inside, the membrane components are going to be recycled back. So these vesicles quite literally are just those phospholipid, the bilayer that kind of pinches off and forms the vesicle with that same phospholipids. So after the vesicle is done, those phospholipids are going to be recycled back. Okay. And then the last type of endocytosis is receptor-mediated endocytosis. Okay. It involves endocytosis and transcytosis of specific molecules. Okay. The cells have receptors embedded in clathrin-coated pits. Okay. So you have receptors within your pits here. Okay. And unlike phagocytosis, you have the pseudopods sticking up, right? In receptor-mediated, you don't have those pseudopods, right? You just have the clathrin-coated pits with the receptors in it, which is going to bring stuff into the cell. So things in the extracellular fluid are going to bind to these specific receptors, which is going to cause the cell to ingest and concentrate specific substances in these vesicles. Okay, so again, not to get to too many specifics, but we can have enzymes using this low density lipoproteins, LDL, cholesterol, iron, insulin, and even things such as viruses and toxins can be taken into the cell this way. Okay, so those are the three types of endocytosis. We also have exocytosis, right, which is where material is ejected from the cell. When it's ejected, it also is created in a vesicle that has that phospholipid bilayer. Instead of just being called a vesicle, though, we call it a secretory vesicle, right? Because the substances in it are going to be secreted through exocytosis. There is a special protein on these secretory vesicles called the snares. Okay, the snares is what's going to allow these vesicles to bind to the cell membrane and eject its contents. Okay, so we're not going to get too far into that. I'm not too worried that you know exactly step by step for that. We'll talk about it a little bit more when we talk about the nervous system. But those snares is what allows substances to be exocytosed. So we have a number of different things that can be exocytosed this way: hormones, neurotransmitters, mucus, and cellular wastes. Okay. All right, and then the last part of this chapter is talking about something called resting membrane potential. Okay. At rest, our cells have this potential across its membrane, okay? So the resting membrane potential is electrical potential energy produced by the separation of oppositely charged particles across the plasma membrane in the cell. Okay, so what it's saying is that you have the cell membrane, right? Inside of the cell, there's going to be a charge. Outside the cell, right along the membrane, there's also going to be a charge. Okay, so the separation of charge gives you a voltage for that membrane, and we call a membrane that has that voltage or separation of charge polarized. Okay, so at rest, all cells are going to have a resting membrane potential that's going to occur right at the surface of the membrane. Okay, so if this is like the whole cell here, right, your potential is only going to be right along the surface. So on the inside of the cell where organelles are and stuff, you're not going to see a charge. But right along the surface, you will see that separation of charge. Okay. So to understand the resting membrane potential and how ions are contributing to this, you need to understand where things are found at rest. So there's a way that I remember the separation of ions, concentration of ions at rest. I think you're on an island. Here's your island. I'll draw a palm tree even for you. There's a palm tree on your island, okay? You're on your island. All you have on your island is a banana. That's a banana, okay? And outside your island, there's your salt water and maybe even a little coconut from the palm tree floating in the water. Okay, so at rest, you have only certain ions on your island or in the cell. So at rest, all you have in your cell is a banana. Bananas are very rich in potassium. Okay, so at rest, potassium has a high concentration inside of the cell. And that's all you have, just the banana. Outside of your island, you have salt water. Salt is sodium chloride. And you also have this coconut, which has coconut milk in it, which I'm just going to say has calcium in it. So I don't know if it actually does, but you have calcium from the coconut milk and your salt from the salt water outside of the cell. Okay, so at rest, inside of the cell, you're going to have a high concentration of potassium. Outside the cell, you're going to have sodium, chloride, and calcium. Okay, and when we talk about the resting membrane potential, it's really important to understand this because these ions are what's creating that separation of charge. Okay, so it's really important to understand at rest, potassium is inside the cell, sodium, chloride, and calcium are outside of the cell. Okay, so your resting membrane potential, how it's maintained. So you have, again, potassium inside the cell. So you're going to have potassium leak channels, okay, in the cell membrane. Again, the leak channels are channels that allow potassium to leak out just passively, no energy required. It's always going to just diffuse down its concentration gradient. So since we have in our cell at rest, potassium inside, right? We have the potassium inside the cell is going to want to leak out of the cell, right? Because out of the cell, there's less potassium, so it's going to just leak out. Also inside of the cell, there's going to be big negatively charged molecules, such as different proteins and different things that are just naturally found in the cell that can't leave. They can't diffuse out, and there's no transport mechanism to get those big proteins out. Okay, so the inside of the cell is more negative. If you have potassium leaking out down its concentration gradient, the inside is more negative. So eventually what's going to happen is the potassium is going to want to come back into the cell. Okay, and that's due to the electrical gradient. Okay, because we have a negative charge inside, potassium is positive, so as potassium leaves the cell down its concentration gradient, it gets outside, and then it's going to want to come back inside because the inside of the cell is more negative. Okay, so that kind of leads to this flowing of potassium out and then back in. But eventually, potassium is going to reach an equilibrium, okay, which means that the drive for potassium to leave is going to be balanced by its drive to want to stay due to that negatively charged. Okay, and that's when we have the resting membrane potential. Okay, when potassium is happy and doesn't want to leave too much, doesn't want to come back in too much, it's just at an equilibrium. Okay, and normally the resting membrane potential, it depends on the type of cell, but around negative 90 millivolts. Okay, and that electrochemical gradient set by the sodium-potassium pump is what's creating the resting membrane potential and this movement of potassium in and out. Okay, sodium can also affect the resting membrane potential. Sodium is positively charged. Remember, at rest, we have a higher concentration of sodium outside of the cell, but we have big negatively charged molecules in. Because of those negatively charged molecules, sodium also wants to go into the cell, right? Because it's attracted to that negative charge. So in some cells, and we talk about the neurons, you're going to see sodium playing a role in the resting membrane potential as well. And those cells will have a resting membrane potential closer to negative 70 millivolts. Okay, the membrane is more permeable to potassium than it is sodium. So potassium is the primary influence on the resting membrane potential. Okay, and we also have chloride, right, and calcium outside. These two don't play a role in resting membrane potential because the electrochemical gradients are balanced, right? The sodium-potassium pump is what creates the gradients and what's going to be creating that separation of charge. Okay, so here's an image kind of showing that. Again, inside of the cell, you have your negatively charged proteins. You have potassium leak channels. Again, at rest, you have more potassium inside the cell. Potassium is going to want to leak out, go to an area of lower potassium concentration. But the inside of the cell is negative, so potassium wants to come back in. Okay, and again, once you have an equal amount of potassium going in and out, that's when you reach the resting membrane potential. Okay, sodium also is going to play a minor role. Sodium is going to want to come in to be close to those negatively charged proteins. Okay, so you have the potassium really driving the resting membrane potential. But the only reason it can do it is because that sodium-potassium pump sets up the concentration gradient, okay? Because we steadily pump sodium out and potassium in, we have that separation of concentration, which allows the resting membrane potential to happen, okay? Neuron and muscle cells use this resting membrane potential and concentration electrochemical gradient to send signals. And when we talk about muscle physiology and nervous system action potentials, you'll see how they use that separation of the electrochemical gradient. Okay, and then the last slide here. We also have some signaling happening in our cells. So these membrane receptor proteins, can also serve as binding sites for signaling in our cells. So we'll talk about this in a lot more detail as we get later on in the semester. We have contact signaling, that cells actually touch each other to recognize the cell's unique surface by membrane receptors on the surface. Okay, and again, we talked about those glycolipids and glycoproteins on the cell surface. The immune cells, right, use that glycolix to recognize familiar cells. So that contact signaling is going to be used a lot in the immune system. Chemical signaling uses chemicals to interact between cells. So chemicals, a fancy word for that we're going to be using is ligands. So ligands are just chemical messengers that are going to allow cells to communicate as well. Okay, so that's it for chapter 3. I apologize for my dog barking. There's a lot of activity out here in Scandia today. So that's it for chapter 3. If you have any questions, let me know.